Right. Vielen Dank für die Einladung zu sprechen. Als guter Europäer musste ich dieses Seminar in deutscher Sprache geben. Aber ich will nicht die schöne Sprache von Goethe, Heine und Koch zu ermorden. Also werde ich Englisch sprechen. In addition, I have a conflict of interest to declare. And that is, I belong to a religious sect known as Genomic fundamentalism, and you cannot expect me to be impartial or sensible. I believe in genomics. And in this sect we have our own creed, our own saying, which is there shall be but one diagnostic test, and that is whole genome sequencing. So don't expect me to give you a balanced view of what I say in the future. So why are we so excited about this? Well, we've had high-throughput sequencing since 2007, and this was over 1,000 times faster, cheaper than anything that went before. It's what economists call a disruptive technology. It's a game changer. It's changed the landscape of opportunities. Several technologies entered the marketplace. Interestingly, we're perhaps now back into a kind of almost monopolistic position with Illumina at the fore. But one other development was the advent of benchtop sequencers a few years ago. So you could actually get a high-throughput sequencing instrument that would fit onto a table like this, fit onto an average bench. And that basically brought sequencing into the average university lab and even into the average clinical lab. Now, the first organism I'm going to say a little bit about, I'm going to mention a little bit about genomics. I know you mainly want metagenomics and microbiomes, but I'll just say a little bit about Acinetobacter baumannii, which I know you have had as a problem also here in Germany. This is a multi-drug-resistant gram-negative bacterium, which is often associated with wound infections, ventilator-associated pneumonia. It can spill into the bloodstream. And in the UK and also in, I think, in Canada and the US, this was a problem in returning military personnel from the Iraq and Afghanistan conflicts. And we saw multiple occasions where you got transmission from military patients onto civilian patients in the hospital. It's got various problems in the lab, hard to identify. And typically what we see in UK hospitals are what we might term serial clonal outbreaks. So someone comes in with an Acinetobacter from somewhere else. It spreads to a few patients, splutters on for a few weeks maybe, and then goes away, and you may be free of the organism for a while, and then another clone comes in and so forth. And you can kind of identify that pattern of epidemiology using conventional molecular approaches, but it's very hard to actually drill down and actually look at the mode of spread and the transmission chains within an outbreak. So I was involved in a study of a multi-drug-resistant Acinetobacter baumannii outbreak that occurred in Birmingham, UK's second largest city, between 2011 and 12. And the first case in this outbreak, as was mentioned typical for many of these outbreaks, was a military patient who'd come back from Afghanistan. I think he actually had flown through Germany on the way, actually. I'm not saying that he got it there. He got it probably out there in the field. It was typed by a reference lab as a unique pulsar type, and we then had an outbreak that went on for 80 weeks and involved 60 patients, including military and civilian. So a really substantial sized outbreak, probably not quite the largest ever recorded, but quite close to it. And we managed to get from our clinical microbiology lab 91 isolates from patients and from the environment, and we genome sequenced them on the MiSeq. First thing we noticed was that, yes, even by genome sequencing, it was a distinct strain. It hadn't been seen previously in any previous outbreak, even locally. And what we did was we managed to, I'm going through this very quickly, but we managed to call out all the single nucleotide variants within the genomes and bunch the genomes together into 7 large genotypes with lots of isolates related to those genotypes by just, say, 1 or 2 changes. And luckily, fortunately, and pleasingly, we actually saw a temporal progression as the outbreak went on, with steadily more SNPs being acquired, single nucleotide variants acquired during the outbreak from the original index genotype. So we're quite pleased that it did kind of make sense and we could actually interpret the data. And when we overlaid the data onto the what we were seeing in terms of time and space, appearance of the organism, which wards and when. Again, we saw a fairly easy to interpret progression. Started off on what we've called here Ward 1, and then towards the end of the outbreak, the second half of the outbreak, it predominantly moved to the burns unit. But the problem was that the burns unit actually has the best infection control in the hospital, because each patient is in their own individual room with their own individual shower and toilet and quite separate from everyone else. So it was a bit of a mystery to understand how this spread was going on. So what did genomics do in this outbreak? Well, it excluded isolates from the outbreak. So we found some Acinetobacter pittii isolates, which the clinical lab wouldn't have been able to speciate. And we could say they're not part of this, not even part of the same species. We were able to, and there were some other Acinetobacter baumannii isolates that weren't part of this clone as well that we found. We could link the patients together by their SNP genotypes, and we could link the patients to the environmental isolates as well. And so one of the key findings was that actually many of the patients in the burns unit had actually been to the same burns theatre, operating theatre. And when we went and swabbed that theatre, we were able to isolate Acinetobacter, and we were able to genome sequence it and show that it was very closely related or identical to some of the isolates coming from the patients. Interestingly, within patients, we saw clouds of diversity. So you might pick one colony from one site and then pick another colony a day or two later, or a week or two later, and you will see that they are just slightly different, a few mutations different. We also saw, though, that patients sometimes have mixtures of completely different clones or different species. So we see Acinetobacter baumannii and pittii in the same sample, or two different clones of baumannii. And typically in the clinical lab, what people do is they just pick one colony that looks as if it's representative of that whole morphotype, colonial morphotype. And this kind of begs the question of whether that really is helpful. What we were doing was largely retrospective, but towards the end of the outbreak, we were able to go from having the isolate to getting the genotype back in less than a week. Now, I mentioned this really just as a kind of set the scene, and it's been written up earlier in the, towards the end of last year in Genome Medicine there, and I'd encourage you to go and look at that. I'd like to move quickly on now to this E. coli outbreak, which you suffered here in Germany, particularly in northern Germany. And some wag decided to call it the sprout break because it was linked to sprouting seeds. I guess it's a memorable title for it. A truly horrible outbreak struck in May and June 2011 with over 5,000 cases and over 50 deaths. And the thing that's quite chilling about this outbreak is that many of these deaths were in people who were young and otherwise fit until the outbreak struck. It's not like some outbreaks you get in hospitals where people are very old and frail. These were healthy individuals. We don't know why, but there was a high risk of hemolytic uremic syndrome, and also particularly females, particularly at risk in this. We don't have a clear explanation. One possible explanation is that real men don't eat salad, and maybe that accounts for the fact that females are there, but we don't know for certain. Now, this outbreak strain was actually sequenced on an iron torrent by the Chinese Genomics Institute. So the guys in Hamburg sent the isolate to them, and they quickly released it. In fact, there was a little bit of a kind of arms race, competition here in Germany and beyond to get the first genome sequence out. And so that sequence was released with this Creative Commons Zero license. That basically the Chinese put it out there in the public domain and said, anyone can do what they like with this. And that led to a load of crowdsourced analyses initiated by my PhD student at the time, Nick Lohman, who went on, because we'd been working with the iron torrent, he actually knew how to analyse the data, how to assemble it. He assembled the data, put out his assemblies, and then invited other bioinformaticians to help make sense of it. And basically, I'm rushing through this because I've told this story many times before, and it's a bit old now, but basically we showed that social media, blogging, Twitter and so forth, could augment academic discourse. And we also showed a key role for this particular new type of benchtop sequence of the iron torrent in that analysis. Now, I've rushed through that because really that's where I want to start. I just want to give you a bit of context. We published that work in the New England Journal of Medicine, along with Martin Epfelbacher's team in Hamburg and people from the BGI. Now, ironically, at the time we did all this analysis, I'd never actually met Martin. And afterwards I said, well, we should meet up, we'll come and see you, and we should have some champagne together to celebrate this success. So there we are, having some champagne together to celebrate our New England Journal paper. And we said to ourselves, well, everyone's going to say this, you're just lucky, you're just a one-trick pony, and you got lucky. How can we actually do something that will make people realise that actually we're quite smart and we can do more than one exciting thing? And so we looked at unique selling points. And I said, well, we're quite good at genomics and bioinformatics and that kind of stuff. What are your unique selling points? And Martin said, well, he's got a freezer full of shit, basically. He basically had over 200 fecal samples from the outbreak. And so, excuse my vernacular, but we decided to start with a project with the title of Let's Just Sequence the Shit. So we just said, let's go and do metagenomics on those fecal samples. Now, why would you think that way? Well, basically, as you know, bacteriology was more or less invented in Germany. There was a small contribution from France and Denmark, but Robert Koch was the main man here. And we've been relying on those techniques that come from the 19th century, even though we're now well into the 21st century. So the question is, can we do better? Or could we do different, at least? So I suggested we take those samples, instead of doing what we did in the initial outbreak investigation, where they grew the organism in the lab and then it got genome sequenced. Why don't we just go directly from the fecal sample and sequence that directly and get the sequence out of that? And maybe we would be able to get the outbreak strain or identify the outbreak strain in those samples. So Martin said, yeah, okay. And they sent us the DNA and we sequenced it. And the very first sample we looked at was very encouraging because we mapped the reads from that fecal metagenome against the outbreak strain genome, and we saw about 25-fold coverage throughout the genome, which was actually, I was quite astonished. I mean, that's not much different from what you get if you sequence a pure culture of an organism and try and get its genome. We also saw some other interesting things there. We saw this kind of smile effect, and that's because the origin of replication in fast-growing cells is overrepresented, so that kind of made sense. And we saw a little upswing where the sugar toxin encoding phage was in the genome, which led us to think that maybe that's overrepresented because there's free phage particles there. But we recognised that JAMA, the Journal of American Medical Association, was putting together a special medical genomics issue, and we thought, well, we'll try them. And we contacted the editor, who then contacted his local expert, which I think was David Relman in Stanford, fairly certain, who said, well, you're kind of cheating, really, because you're aligning your reads from your metagenome against the genome of the outbreak strain. But in an outbreak situation, you wouldn't know what the outbreak was caused by, and you wouldn't know what the outbreak strain genome looked like. And it's a bit like trying to solve a jigsaw puzzle by just assembling your jigsaw using the lid, the image on the lid. Basically, you've got to try and solve the whole thing from scratch. So luckily, Nick Lohman who was working in the lab at the time, he was up for the challenge, and he did a few all-nighters working on this. And this is one of his first plots. He said, right, let's look at, we'll assemble these metagenomes, and then look, we ended up with, I think it was 40 samples we got. And he said, let's have a look at the taxonomic complexity that we can see when we look at these samples. So here are sequences that are present in at least 2 samples. And you can see it looks like a taxonomic mess here. You've got it splayed out according to GC content, which is a way of distributing bacteria taxonomically, and we got the coverage there, and he did a quick taxonomic classification, and you can see it looks like a mess. There's the Enterobacteriales are there, which has the E. coli outbreak strain in them, but trying to work that out, separate that out from the rest of it, you can't see any easy way of doing that just looking at that. But Nick's a very bright guy, and so he thought, well, one thing we could do is we could say, well, if this is an outbreak, and let's say that these 40 samples we have are kind of representative of the outbreak, then we should expect to see the outbreak strain sequences in at least half of these samples. So let's just throw away everything that's not present in at least half of the samples. And you can see that when he did that, he did actually achieve a considerable degree of taxonomic simplification, threw away a lot of the data that we didn't want. But we still, and we have got the Enterobacteriales looking more prominent, but we're still a long way from having the actual genome sequence of the outbreak strain. And then he made one more leap, intellectual leap, and thought, well, how can we get further? He could subtract all the sequences that are found in normal, healthy people. Now, luckily, he didn't have to go out and wander the streets of Birmingham to find 40 healthy people and ask them for their stool samples, because the MetaHit project had already done that in Germany, and there were many examples of stool microbiomes from healthy Germans that he could use as a comparator group. So that's what he did. He chose a series of age-matched controls from that data archive and subtracted everything that was in those from healthy individuals, and ended up basically having the genome of the outbreak strain. Those of you who are quite sharp will say, actually, he hasn't got the genome of the outbreak strain because you've subtracted all the stuff that's in E. coli as the core genome. Yeah, he actually got the accessory genome of the outbreak strain, and then he used another clever approach, which I won't go into, to reconstruct the actual full genome. And so we ended up getting that published in JAMA, and we're very pleased that we managed to get a second paper out of this sequence, this sequencing. How am I doing for time? I've got a little bit more time. So I'm just going to quickly shift over to ancient DNA, where we've looked at this in over recent years. There have been some spectacular successes with that. Living here in Düsseldorf, you must know about the Neanderthal genome sequencing project. There is me this morning with one of my newfound Neanderthal friends and his genome in the background there. But there is obviously a problem with this kind of approach, and there's a risk of contamination. And there have been some spectacular failures. So the very first Neanderthal sequences reported in the literature, this first megabase of Neanderthal sequence, 8% of it was actually modern contamination. And it's actually really quite a vicious field, ancient DNA work. People are always dissing each other and saying, that their work is rubbish. There's this kind of skeptics versus believers. For Egyptian mummies, there was a paper in JAMA where they said they got genotypes from Tutankhamun and his family, and then a second publication a few weeks later saying this is all rubbish, absolutely not possible to get DNA out of ancient Egyptian mummies. Well, if you're looking at pathogens, there is quite a literature now on ancient DNA work on pathogens, and perhaps the triumph there was the paleogenomics of the Black Death. And clearly this is a field where there's less risk of contamination of DNA from the handler or the environment. So if you're looking for TB or Yersinia pestis, it's unlikely that the handler is going to have tuberculosis or plague and introduce that into the samples. But there is still this risk of contamination. There's still this skeptic versus believer schism in the field. So I was approached by this lady, Helen Donoghue, who had some samples from 200 years ago, lung samples, and she had found evidence from PCR that these were TB positive. And they wanted to see if they could get any sequences out of them. And I said, well, we're not going to fiddle around doing any more PCRs. We'll just do shotgun genomics, building on our success with the E. coli outbreak. And it avoids all the problems of amplification, where you've got carryover, but it also avoids the problems of a popular technique, hybridization capture, because you don't quite know what you're going to find in a sample. And if you use shotgun metagenomics, you'll find anything that's there. Whereas if you design probes, you're only going to find what you're looking for. And also, it's a lot of effort and a lot of faff to design probes. Now, we sat in our chair designing the experiment. We thought, well, it probably won't work. There'll probably be too much human DNA. Any TB DNA will be swamped by that. And there'll probably be all these mycobacteria that come in after post-mortem. But we did the experiment anyway. The samples she gave us were from Hungary, from a crypt that was discovered in 1994 that had over 200 bodies in it. It had been sealed up since the early 19th century, and many of those bodies have become naturally mummified. And so we focused initially on this woman here, Teresia Hausmann, who died on the day after Christmas Day in 1797 at the age of 28. She had a clear chest X-ray. They basically investigated these mummies extensively with X-rays and other things. But she did appear to be cachectic, which is consistent with TB, and they actually did microbiological analysis, even did ZN staining on her tissues and molecular staining such as PCR, suggesting that she had a good quality of DNA, of TB DNA in her sample there. And the interesting thing was that we actually have the death record there, so we know exactly which day she died, so that kind of gives us a chance to date these things. So what we did actually, we started with her, but then we went on to look at a wider range of individuals, and we ended up getting TB genomes out of 8 individuals from this collection. And we were quite astonished that we were able to get coverage, sometimes going up into the hundreds for some of these samples, which was just a staggering. And using a high-seq, even in ones that were had fairly poor yields of TB, we were managing to get up to nearly 1-fold coverage. And what we found was that all of the genomes, these 200-year-old genomes, they all had a particular deletion, which meant they belonged to one particular lineage of TB, the so-called Euro-American lineage, or lineage 4. And interestingly, what we found was that there were mixed infections, so there were mixtures of genotypes in these bodies. Now, when I first heard about this, I thought there must be some mistake here because, you know, mixed infections in TB, that's not something I'd heard of before. But we actually looked at the literature and we thought about it. So basically, as I mentioned when I was talking about Acinetobacter, the conventional practice with TB is you pick a single colony and follow it up. So if there were a mixture of 2 or 3 or 4 strains, you probably wouldn't know it in the lab. And there are plenty of case studies in the literature saying that you can get mixtures. You can get mixtures of resistant and sensitive genotypes in the same patient. And in one study in KwaZulu-Natal, they said that up to 1 in 5 cases in that high prevalence area had mixtures. So basically, it made us rethink about the way in which we do TB diagnosis. Now, we were also able to use the sequences we had to place these genotypes on a large phylogeny of 1,500 modern genomes from this particular lineage 4. And that was quite a nice finding that we could actually place them there and see that they were distinct but belonged in different places on that tree. We found that Teresia Hausmann and her mother had a pair of genotypes that were the same in both mother and daughter. So that's very interesting, first tight epidemiological link between 2 long dead individuals with TB. We also did some phylogenetic dating and using the program BEAST, and we said that probably the most recent common ancestor of this whole lineage was in the late Roman period. And this was consistent with a relatively recent origin of the whole TB complex. And this is a very controversial point at the moment. So Johannes Krause here in Germany, been working with people and come up with this idea that basically the TB complex is only 6,000 years old. Other collaborators of ours in Israel have said, well, they've got Neolithic TB samples that are, I think, 9,000 years old, so that can't be true. And so we're at an interesting stage where there's a conflict between what some of these genomic analyses are telling us and what other people are saying. And it says need for genome-level evidence from Neolithic samples, and later this month we will be receiving some Neolithic samples from Israel to actually go and look for genomes in them, so we'll be able to tell. So this is published in February, so you can look at that if you're interested in Nature Communications. How am I doing for time? I suppose, yeah, I've still got time. I'll just tell you another ancient DNA story. So we were working with an Italian physical anthropologist, Raffaella Bianucci, and she gave us some material from a skeleton from Sardinia that had been buried in the second half of the 1300s in this grave here in an abandoned medieval village. So the village was abandoned in 1426. And when they excavated the skeleton, they found these calcified nodules in the pelvis. And it wasn't clear what those calcified nodules were, but we got given them, and they said, you can have a look at them, see if you can get any sequence out of them. Now, I guess anyone who's a clinical microbiologist in the room will say, oh, calcified nodules, I bet you're going to tell us you found some more TB. And that's what we were thinking we might find if we were going to find anything at all. But the advantage of using an assumption-free shotgun metagenomics approach is that you're not actually tied down to one particular organism. And what we actually found was Brucella. So we ended up with a 7-fold coverage of a medieval Brucella genome, with the 2 chromosomes there showing generally even coverage of those genes, of those chromosomes, and also showing some signs of DNA damage consistent with this being aged DNA, which is shown there as well. So that was very interesting. We went on to be able to genotype this genome and put it onto a phylogenetic tree with modern brucellas, showed it was clearly belonged in the species Brucella melitensis. And in contextualizing this, we can say that it's clear that there's been brucellosis in Italy for a long time. So the skeletons at Herculaneum actually have bony signs, many of them, of brucellosis. But there's only been one other detection of brucellosis from ancient DNA research, and that was in the 10th or 13th century Albania, and that was just done by PCR. This organism was acquired from sheep and goats, and there's a long history of sheep and goat farming in Sardinia, so that kind of makes sense. We don't know whether this individual skeleton 2568 was a farmer, and that's how he caught it, or whether he just ate dairy produce. He has this other condition associated with the bony pathology called DISH, and that's supposed to be associated with sedentary lifestyle, so maybe he was just someone who ate the dairy produce rather than being a manual labourer, but we don't know. We've done other ancient DNA research. I'm not going to go on about where we've looked for signatures of the Neolithic transition in submerged sediments, and we found wheat, evidence of wheat in the British Isles much earlier than people were expecting. But for those who are getting a bit bored and saying, well, what are you doing all this ancient DNA stuff? Well, the kind of irony is we did the ancient DNA stuff on TB through metagenomics before anyone had ever done metagenomics on contemporary TB samples. We said, why don't we just do that? And so in collaboration with our colleagues in the Gambia, in the Medical Research Council unit in the Gambia, we did actually take sputum samples and we looked to see if we could find TB sequences direct from the sputum samples without having to grow the organism. And this was done by my PhD student Emma Doughty, you can see there in the picture. And here are just the headline figures of how many reads we got and how many aligned to the human genome. So in some samples, here we were swamped with human sequences. But in all of these 8 samples we looked at, we did manage to get some sequences that aligned to the reference strain of TB. So we could say, yes, we've detected TB sequences in that sample. And interestingly, for most of the samples, all bar one of the samples, there was enough information in the sequences we were recovering to be able to place them on a phylogeny of TB. And so we could say, okay, so most of them belong to this Euro-American lineage, but there were 2 of them that belonged to a West African lineage. Now, unfortunately, we didn't get high enough coverage to do anything like looking for resistance-associated mutations. It wasn't, so we're not saying that this is the final solution, the obvious way in which you should do TB research in the future. But we were very pleased that we just even achieved proof of principle, and we're now trying to optimize the extraction procedure so that we get more TB DNA and less human DNA when we do this in future. So where are we going with this? What's the future hold? Well, obviously one of the interesting developments lately is this nanopore sequencing, Oxford nanopore, with these devices at the size of a USB stick, and you just plug them in your computer and the data streams off in real time to your computer. I think there are various tipping points. In England now, whole genome sequencing has become routine for TB. So all the TB isolates get their genome sequenced now. And just last Friday, I tweeted on something I just heard, which is that in England also now all the Salmonella isolates are now getting whole genome sequenced, and they're not doing phage typing anymore. They will rely on SNP-based genotyping in the future. Metagenomics, well, is it going to work in the future? It was a very unusual situation where we were doing it, and we were doing it in retrospect. There have been a few other cases, I haven't mentioned them, some of them in Germany here, where people have used it for diagnostic emergencies. We've got a sick patient, we don't know what's going on, take some tissue samples and sequence them. And so that, I think, is a valid way forward. Whether we'll be able to expand this to make it routine in the lab, I'm not so sure. It's very hard to predict the future, but it's possible. And we still, I think the idea that you can use metagenomics everywhere is probably naive, because in some samples, say in a blood culture, there's such a small amount of bacterial DNA. There may only be a few cells there. It's going to be very hard to detect that by metagenomics. Maybe not impossible, but I think we have to be aware of the limitations. I think one of the things that's interesting is that we can speculate in the future that perhaps we're just going to collect macromolecular data from patients and just sequence all of it. And then the analysis will be downstream in terms of the bioinformatics. And there's a lot, microbiology here is kind of running in parallel with a lot of other disciplines. So people are very interested in circulating DNA for looking at tissue rejection in transplants, looking at progression of cancer and so forth. So I think we've got an interesting avenue, particularly if this nanopore sequencing does become a lot easier, cheaper and more reliable. Kind of obliquely quoting Karl Marx there to say, actually, I'm not saying anything about microbiomes. I could have said a little bit about chicken microbiomes, which we've done some work on. But the point is not to just to understand these metagenomes and microbiomes, but actually to change them. And I think an interesting area for future research is actually going in there and reprogramming microorganisms to go and influence those microbiomes. So go beyond just natural probiotics like the Nestle strain that was developed here in Germany, and actually go in and actually make things that are much smarter in the future. And that's another little quote there at the bottom in German from the Neanderthal Museum as to where we're going. One last thing I'll say is that one of the other conference organisers mentioned this, that one of the problems is actually doing the computer analysis. And we have this problem of what some people are called drinking from the fire hose. Basically, we've got this big data issue. And one of the solutions that we're exploring in England, well, in England and Wales, is cloud computing, so that we can actually provide a bioinformatics infrastructure that's very useful for lots of people in lots of different universities in a distributed fashion, but allows easy sharing of information and easy sharing of pipelines and makes things a lot more reproducible. So that's a project we've got ongoing at the moment. So in conclusion, there's a quote there from Lincoln Steffens, who is an American journalist, who I first used that quote when I was in Berlin, not far from where the Berlin Wall used to be. Lincoln Steffens comes from the days of the 1920s when he went to the Soviet Union and he went back to America and saying, I've seen the future and it works. So there's a kind of irony in that statement because the Soviet Union did kind of work, but not forever. Another phrase there from the guy who invented cyberspace and cyberpunk, William Gibson. The future is already here, it's just not very evenly distributed. Yogi Berra, who died recently, made this quote, predictions are difficult, especially about the future. So I don't quite know where we're going, but it's quite fun to explore this. And I'd just like to acknowledge all the people who've helped me along the way. I won't read their names out because time is running out. But that's it. Thank you very much.
Thank you very much for this fascinating presentation. We have time for discussion. Colin.
Hi.
Could you see a future for developing?
Just realised I didn't have my microphone on, did I? The whole
Could you see a future for developing countries where the infrastructure for classical microbiology is actually just not there?
Yeah.
It's not going to be there for quite some time to come, but they could in fact use this kind of technology.
Yeah.
Getting a little bit of ability to sequence is actually not difficult.
Yeah. Yeah, I think that's the great hope for the future. My former PhD student and now collaborator, Nick Lohman, has actually been in West Africa using nanopore technology to sequence Ebola isolates there. And they've basically gone out with all the stuff they need just in their hand luggage to go out and sequence those Ebola isolates. It's hard to know, nanopore has actually been hyped up for the last several years, and people are still starting to get a little bit impatient. When is it really going to arrive? And people are saying it's not going to be an Illumina killer. We have been talking to people in the Gambia about whether they should be getting sequencing going. They've bought themselves an iron torrent, but they can't really get it to work, and it doesn't work that very well. And they're hoping to get an Illumina. In South Africa, they've now moved on to genotyping all their TBs by whole genome sequencing. So I think these technologies are accessible and they are moving progressively outward. in recent months, we've collaborated with people from Al-Quds University in Palestine, which is kind of middle-income country. We've got a collaboration with some people in Lima, sequencing TB isolates from Lima. So I think we are, these technologies are much more accessible than anything we've seen before. And in our lab, with the Illumina MySeq, we basically let anyone who's even vaguely competent use it. So PhD students, project students, postdocs all use it. Dozens of people have used our Illumina MySeq, which is by a great contrast from, say, 5-6 years ago when we had a 454, and we had just one individual who was trained to use it, and nobody else could get near it because it was so difficult to use and so expensive if anything went wrong. So we're in a different place now, I think.
Please.
Hello. So I'm having two questions.
Yeah.
The first is, did I get you right that it's done routinely in the UK that every new TB strain gets whole genome sequencing?
That's correct, yeah.
Okay, so how more sensitive are you then for detecting the resistant ones as compared to the normal hybridization assays or what we all perform for resistance? Because they're quite good for isoniazid and they're just really bad for the second line.
Yeah, exactly. I think that it's a work in progress. We have been looking at some West African isolates and some collaborators in the Gambia and in London. And as you say, it's quite easy to call out the most common resistance-associated mutations for the common drugs. But when you're getting down to ethionamide and caprimycin, and it's much more of a guessing game. We don't really have enough of an evidence base at the moment. But the problem is that even the conventional approaches are not great. They're not particularly good and not particularly reliable. And so it's hard to know what is your gold standard. So just yesterday we were having a Skype discussion about the fact that with the ethambutol resistance we were getting some discrepancies where things were, by conventional approaches, we're looking as if they're sensitive, but they were having some resistance-associated SNPs in the genome. And we were saying this is probably not a mistake that we've made, it's just the way this technology is at the moment. The conventional approaches are not great. And they obviously take a long time to do. You have to take another couple of weeks at least from having a pure culture to go and do the sensitivity testing. So, I think for the conventional easy stuff, rifampicin, which is obviously the most important thing, I think it is fairly straightforward and they're reading it out. There's some guys in Oxford, so much of the pioneering work was done in Oxford, Derek Crook's kind of empire there, and they have developed, there's a guy, Zamin Iqbal, who's developed an assay, a website and a thing where you can just drop your genome on and in real time in 10 or 15 minutes it then spits out what the resistances are. Mm-hmm. And he's been validating that and I think he's getting good results.
Okay. So the second question is, what do you think about this, let's call it preventive metagenomic sequencing of fecal content? So the idea of a lot of people is that you could get information about possible extensively resistant bacteria patients colonizing with beforehand against immunosuppression or something like that.
I think it's a great idea, and I wish that I could persuade funding bodies in the UK to give me some money to do it. I mean, I have tried to put in grant proposals to look at intensive care unit patients and just follow their fecal microbiomes, particularly the long stay ones, to see what happens. There haven't been many papers, peer-reviewed papers on this, but the strong presumption is, I mean, Eric Payne's done some work on it in the US, but the strong presumption is that patients who sit in the intensive care unit for a long period of time, they have multiple antibiotic dosing, and their microbiota does become simplified, becomes more vulnerable, and it probably becomes more susceptible to being invaded by multi-drug-resistant organisms. And then when those organisms sit in the bowel for a long period of time, they reflux up, and then they'll eventually will cause ventilator-associated pneumonia. They may get into the bloodstream, but there probably is a window where the patient is colonised before they actually get disease. And I think that like you said, I think there are good reasons for taking that seriously and having a look. But we don't know for certain until we've done it. We have to do the experiments, collect the evidence, and see. So yeah, I think it's a good idea.
the question as well. The first question is, we always think that resistance is new, but I mean, all the bacteria have evolved some strategies to have antimicrobials to have an advantage in growth.
Yeah.
The question is, with the microbacteria that you found in the 18th century, are there any mutations already there which give resistance to modern antibiotics, or are they totally sensitive?
They're all totally sensitive. I think that you sometimes do see the resistance genes when you look in old material, that actually are carrying beta-lactamase genes, things like that, on the transposons. I'm not aware of anyone showing. Because TB is quite an unusual case where it's all mediated by single base mutations in the chromosome. I'm not aware of anyone showing that those kind of mutations are out there long before people started using modern antibiotics, and it would be a bit surprising if it were true.
And my second question would be, always sequencing is the basis, but then you need all the bioinformatics. And
That's
what
true.
would be the setup that you have to cover that? So are you doing that all in your own lab?
So all the work that we've been doing is in our own lab, and there are dynamic tensions inherent in this process. We kind of slowed down a bit, but over the last few years, the sequencing has just changed. Every few months, a new kit has come out, getting longer read lengths, you're getting new ways of making your libraries, and if you're in a research lab, you can just say, let's just take the latest thing and do it. Yeah, just keep chasing that advancing wave. If you're in a clinical lab, what you want are standard operating procedures that everyone agrees on. And it's very hard to come up with standard operating procedures that when everything's moving fast forward. We used to joke that, Public Health England, our public health body, they were stuck in the 20th century. They were never going to get there. But in the last couple of years, 18 months, 2 years, they really have changed their game, and they really are doing this, and they are actually grasping this and saying, okay, well, we can't sit about and philosophically worry about whether we're going to get the perfect pipeline. We'll have to just get on and do this. And we've got problems ahead because within the EU, for various pathogens, the way in which you type them are actually written down. The regulations are agreed. So things like phage typing is kind of a standard. And to say, well, we're just going to do it all by genome sequencing upsets those who like to make the rules. And so I think there are some interesting questions ahead of us there as to whether for TB you can get rid of spoligotyping and myro-VNTR across Europe, whether you can get rid of phage typing for salmonella. So these are interesting challenges.
Do we have a second question? Third one, actually.
So I also like the sequencing approaches a lot, and we do them in our lab as well, but I don't know whether you saw a paper by Alan Walker.
Yeah.
Where he just nicely showed that depending on the kit you're using,
the ketone, I know Alan quite well.
The room you're sequencing, the person who's doing it, you get totally different results. So what are your strategies to
So his concern is particularly with 16S. So when you're doing molecular barcoding, and I would say 16S is a 20th century technology. Okay, David Relman gave it a push forward when he showed you could use it with high-throughput sequencing as well. But there are some problems with 16S in terms of what you get in the kits if you're not careful. Often things are there in very small amounts, and because you're using an amplification-based technology, you can find them. And Alan wrote that paper, basically said you're getting things. So you've seen the papers on the brain microbiome appeared in PLOS One. There was a paper on something called Bradyrhizobium enterica, which annoyed me, not just because it was wrong, I can't believe it, but they don't know their Latin either, because it should be Bradyrhizobium entericum. But there's a lot of nonsense out there, and I think we have to be careful of that. Some of those things, if you just did shotgun metagenomics and there were small amounts of contaminating material, you might find it there as well. It's possible, but you're not amplifying things, so hopefully you're going to get a slightly better representation of what was in the starting material. But there are issues that Alan's brought out about the way you store samples. He says that if you take a stool sample and you analyse it freshly, you'll get a different result from the same stool sample that's been stored in the freezer for, say, a year. He said that Bacteroides, even though we think at -80 everything must be inert, you get that the Bacteroides DNA does seem to degrade, and you'll get different results. And he says that one of the concerns that you have to be aware of is that if people say there's a difference between this population and this population, have they actually analysed those two samples? I mean, we could be guilty of that, obviously, when we did our study where we took information from somewhere totally different, where it was prepared in different ways. But that's the issue, that you have to be very wary. But this doesn't mean we can't progress. I mean, I'd like to call myself a sceptical enthusiast. You have to be sceptical about everything. And any of you who've been looking at nature will know that our claims about Neolithic wheat are currently under the spotlight, and people are saying, is this real or is it modern? Is it really ancient? But we have to progress.
Are there more questions from the audience? Not that I asked a more philosophical question, maybe. Okay. With all the sequencing technology now, if you look on the microbiome of an individual, how stable is that? If you look over years or months?
I haven't done a lot of work on the human microbiome, but basically the accepted assumption is that it is fairly stable in the individual, and it's stable if in the absence of any perturbations, it's stable in the individual for years. We know that giving antibiotics, though, does have quite an effect, and that can be quite long-lasting. And one of the things that puzzles me is that we have this, basically the word on the street is that the microbiome is stable, and there is this phenomenon of colonisation resistance, which means that it's very hard for anything new to come in. So you've got an E. coli in your bowel, a new E. coli can't come in. But one of my former colleagues, Peter Hawke, has shown in the West Midlands of England that large numbers of people who go to Pakistan or India and then come back with ESBL producing E. coli in their bowel. So how did that manage to get in and elbow out the other E. colis, or is it just sitting alongside the E. coli that was already there? I think there are interesting questions about this stability and what colonisation resistance is and how it's overcome. And the question about what happens in critically ill patients in hospital, I think, these are interesting questions.
It's all open to.
It's all open. There's lots of. It's not a closed field. There's loads of research left to be done. We just started.
Okay, thank you very much. That was really fascinating.
Thank you.